Dismiss. Okay, my recording has started. Okay, so let's go for this downstream analysis with cluster. So I'm going to talk a little bit about general concept about data mining, clustering approaches for RNASeq data, and then tools for performing clustering analysis. Clustering, let me tell you that, of course, this is one of the things that is a whole universe by itself, okay? Clustering is a part of the thing that is called non-supervised models, okay, with that of data mining. And as I said, it can be whole courses of statistics if you want. So it's difficult. So I have been trying to synthesize more or less the main ideas and put it here. So let's start with this general concept of data mining. So when you are doing data mining, anything, basically the thing that you are doing is trying to discover new patterns. No, it's the concept of data mining is trying to discover new patterns. And we have been using data mining for everything. You can take some statistics of populations and use it. No, I think that the most recent example of data mining and how the data is used is Cambridge Analytica, and how Cambridge Analytica was influencing votations, okay? So you can use data mining for everything. I remember that when I was at Virginia Tech, I was using data mining for a simple thing that was not related with science. That is, we were developing new programs for students, no? So we wanted to develop a new plant science program for the students. And they were looking for new names for the classical topics, no? In the US, at some point you change the names of plant physiology or plant pathology, and you put a new name that is health of plants. So I remember that after a big discussion in a table about how we should call some courses, or how we should call the degree, the thing that popped up was, okay, why instead we decide how we should call something, we run a data analysis, we run a data mining. And the thing that we are going to do is we are going to go to all the job databases that you can find in the US. We are going to retrieve all the jobs that has the plan descriptor. And then we are going to mix all the words about the requirements and about the knowledge that they are asking in these companies. And with that, we are going to develop what the thing that the student needs in order to fit what the courses that they need with what are the companies is asking. No? Because as I said, if a company is asking for a plant physiologist, but then they don't use that name, they use a different name, okay? They use, I don't know, a botanist or general botanist or something like that. We needed to know that. So of course, we download all the data of these databases, we run this analysis, and we pop up with several terms. That was the terms that we were using to develop the program. As I said, nothing related with science. You can use data analysis for anything that you want, as soon as you have data and then you have the models to run the data. And in my case, I didn't run any complicated model. I just ran one of these cloud analysis in which the most important terms will highlight. You know, that is a good example. That will be a good example of summarization, okay? But you can use anything. Okay, so when you have a data mining, you can divide the data mining in four different types of data mining. You can find for an anomaly detection, you can look for outliers, changes, or deviation of the detection. In some ways, something that doesn't fit. When you are using RNASeq data, outliers are good sometimes. Outliers is something that they don't like at when you are running a test, okay, when you are running all these differential expression analysis and all these tests, because you expect a distribution. So outliers doesn't fit very well in the distribution. But in some ways, outliers for us could be interesting because help us to see that the experiment is running well or not. And the most classical example of an outlier when you are doing an RNA-seq analysis is Rubisco in plants. When you are running a transcriptomic analysis with leaves, you should see all the genes related with Rubisco in a high level of expression because it's one of the most abundant proteins in leaves. Okay, so you should see that as an outlier. So that means that everything is correct. In other words, if you are, let's put the example that you mix the labels and you are working with roots and leaves, and then looks like in your roots, Rubisco is the outlier. No, that means that there is something wrong. So outliers are good because help us also when you know, because maybe you don't know what are going to be your outliers, and when you know your outliers, helps you to understand the data. Then you have this association rule learning, that is dependency modeling, that we don't use in RNASeq at all, because they search for relationship between variables. So it's not, you can maybe use sometimes in ecology, but as I said, we don't use in RNASeq. Then we have the two ones that's the unsupervised and the supervised method. The unsupervised are clustering, and the supervised are classification. And both of them, the thing that you do is you put things in groups. So for the clustering, the thing that you do is you group things based in spatial profiles, for example, you will see. And with classification, you have some kind of manual curation or semi-manual curation in which you put things in boxes that can be, for example, metabolic pathways, okay, or GO terms, as you will say. You also have regression, we are not going to see that. And then summarization, as I said, including visualization or report generation, or as I said, this cloud of words will be a good example of summarization. So as I mentioned before, for gene expression, the thing that we use is clustering, classification, and summarization. It's kind of the most important things that we use with transcriptomics. Okay, being said that, let's go to these clustering approaches. So, of course, I'm not going to tell you these things about how you can have your with data mining things, no? So that is something that this is out of the program, okay? But as I said, keep in mind that this is a very, very useful thing. If you didn't see, there is a really wonderful documentary and scary documentary in Netflix about Cambridge Analytica. I strongly you to recommend you to watch that documentary if you didn't see it. Okay, so what is clustering? So clustering analysis of clustering is the start to assign a set of objects in two groups, okay? So the objects in the same cluster are more or less similar in one sense or another. Similarity, of course, is something that can be defined in a lot of different ways by itself than any other in other clusters. So they are considered unsupervised machine learning methods, okay? So for example, if you have a cluster of things, a group of things, then you can start to group them by similarity. And then you can say, okay, so this is cluster 1, cluster 2, cluster 3, and cluster 4. So one of the characteristics of clusters is that you are generating new information, analyzing the data that you already have. And that can be something interesting. So clustering is a main task of explorative data mining, and the most used, okay, the most used, that doesn't mean all, but this is the most common ones, used for gene expression are principal component analysis, hierarchical clustering, and Cummings. Okay, so let's go to explain each of them, as I said, without entering too much variation. So if you don't know, I mean, principal component analysis is one of the things that basically we use a lot in a lot of different type of data, and it's the first thing that you should do with your data. We already do principal component analysis when we were looking for the relationship between samples. And basically the thing that you do is you reduce the dimensionality of the data based in their structure. So you have three dimensions for your data points. The first thing that you look is you analyze your data points and you try to look for the variance maximization. You start to combine, you combine these three components. You measure then the variance of those, so it's a transformation, and then you maximize the variance. And then you maximize the variance in one order, then you maximize the variance in another order, and you maximize the variance in as many components as the initial variables that you have. So once you have do that, then you take the ones that explain you take the first two, for example, that explain most of the variance, and then you represent your data there. So this would be the principal component one is this one that is contain the maximum variation, and the second one contain other part of the variation. Okay, so that will be in some ways in a very rough way to explain what is a principal component analysis. Of course, you have all these mathematics behind it. Okay. The principal component analysis can be used for data exploration and data visualization. I really use a lot. So we use principal component analysis not only for RNASeq data, but also when we are looking for genotypes in a population. It's always good to have this principal component analysis to see the relationship between them. So as I said, it's a good way to have this data exploration and data visualization. When you are performing principal component analysis, you need to keep in mind several things. So you need to consider the normalization that you are going to perform, data filtering, the outliers that you may have, and also the interpretation of the results. Okay, as I said, I don't want to go into much detail there. Of course, as I said, you may have run at this point several principal component analysis. So looking into the clustering, as I said, probably one of the most important ones is the hierarchical clustering, or also called HCl. So the distance between the elements, and you can define distance as distance between genes, genetic distances, or whatever distances you want to use. So you use these distances to build the cluster. No? So the thing that you do is you have a series of elements. So you calculate the distance between them, and then you start to group them by pairs. Okay. So you say, for example, these two are together, these two are together, these are two together, together, together. And then once you're starting to have this by pairs, you're starting to group again by pairs, one and again and again and again by pair of groups until all the elements have been assigned to groups. Okay. So that will be the method. Of course, you have some mathematics behind, no? Because mainly you have two things that you need to think about it. First is how the distance is calculated, and then how the linkage of these groups, the agglomeration, is performed. So how the distance is calculated, there are different ways to calculate the distance. You can use Euclidean, score Euclidean, Manhattan, maximum, Mahalanobis, etc., etc., etc. Which one you should use? It depends. So by default, we are using Euclidean because it's very simple. But of course, there are some other ones that can be more suitable for your data. And how the linkage is performed, how the agglomeration is performed, again, it depends. You have complete linkage, single, and weighted average linkage clustering, that is this UPGMA, weighted average linkage clustering, that is this WPGMA, cycloid linkage clustering, minimum energy, work criterion, etc. The ones that we are using in most of the RNASeq analysis is work criterion. That said, sometimes it's not good, but as I said, we are using usually that is the thing that you have in some of the tools by default. So how you need to select this right criterion? It depends, no? Because for example, when we are looking for these agglomeration methods, some of them have some advantages compared with other ones. So for example, single linkage has the advantage of the number of clusters that you get, but then you have these like cliffs that may have problems. Complex linkage has compact classes, but one observation can alter the groups, so that is another problem. Average linkage, you have similar sizes of variance, but are not robust. The centroid has robust to outliers, but then it has a smaller number of group clusters. Or have minimized this inertia that you have for some of the groups, but then the class you have a small number of classes if you have a high variation, no? So this is, imagine that you have this data and you need to do this kind of analysis. So this is an example of single linkage, no? You go to the closest. I'm not going to the math here, but as I said, remember that you have some math here. The complete linkage look for the maximum distance. This is the shortest, this is the longest. This is the one from the centroids. This is an average between all the elements. And the word, as I said, has another transformation by itself. Which one you should use? It depends on the clustering goal. Okay. Sample or genes. So you have the samples, the target to cluster them by the overall expression. You may use some. If you want to cluster them by pattern, you may have another one. So, for example, by overall expressions, things like word methods could be useful. Meanwhile, for patterns, average linkage could be more useful. Okay, it depends. So that is in terms of hierarchical clustering. Then you have this Cummings clustering. So this is an interactive algorithm that's trying to partition the data sets in a number of groups. So with hierarchical clustering, you don't define the number of clusters. You're starting to classify them, and then you have a tree. At some point, you can take this tree if you want. I will show you that thing later, and take a number of cluster X. But you don't have a predefined number of clusters. Meanwhile, with Cummings, you predefine the number of clusters, and then you try to assign all these groups to this number of clusters. Okay, so that is the idea here. As I put in here, no. So the thing is, it tries to make inter-cluster data points as similar as possible, while keep the cluster as far as different as they can. So usually the thing that works is how it works is you have 3 steps. So the first, you specify the number of clusters that you want to have, and then starting to calculate the centroids, first shuffling the data sets and randomly selecting K data for the centers without replacement, and then keep interacting until you don't have changes in the centroids. No? So you try to maximize the distance between the clusters. As if you want to have a good video, I think that you can click here. They have a video about the interactions. I think that is very representative about how this method works. Basically, the idea is that, as I said, no, you have as a group of points that is represented by these squares. Okay, so you represent centroids randomly, as you can see here. And once you represent the centroids randomly in your space, you say, well, you assign the groups. As you can see, no? This square will work with this one, this will work with this one, and this will work with this one. But this is the thing that they call Voronoi diagram. Then you move the centroids, okay, based on your data. So for example, this will move here, this will move here, this will be moved here, and then you reassign groups, okay? And you repeat this once and again and again and again until you reach some stability in which that doesn't change anymore, okay? So that will be the process. Of course, this can be complicated. So my recommendation is there is a good review that I found when I was preparing the class, that this is avoiding common pitfalls when you are clustering biological data. Okay, and that was a very useful review. No? So let me summarize what is the important messages here that you should have in mind when you are going to try to do clustering on your data. First is you need to know the structure of your data, the underlying structure of your data. And to know that, probably to do a PCA would be a good idea. Then it's important to know about the dimensionality of your data, because as I said, you may have one axis in which you don't have many samples, and then you have another axis that can be massive because you have genes, okay? Also, how many relevant features of the biological questions you are asking. So that is another important point here. And also, and this is essential, the noise and the variance of the data. If your data is very variable, you have a high variance, and you have a lot of noise, this is going to be difficult in order to cluster your data. Each of the algorithms, as this says here, have some assumptions, okay? And then it may give you different results. The 4 classes that they are spelling here, even if I put 2 examples, are hierarchical, centroid, density, and statistical. I explained this hierarchical and centroid, but let me give you some ideas of those, okay? So So the hierarchical clustering is useful when the data that is expected contains cluster with clusters within cluster, or the observations are expected to have nested relationships. Okay, so when you are thinking that you have nested relationship, that means subgroups inside groups. Okay, that could be a good way. And as I said, the WAR algorithms can produce different results depending on the threshold of similarity, highlighting hierarchical relationships. So WAR is the thing that you should use, and depending on how you use it, still you may have different results. Centered clustering, like Cummings, assign the cluster based to the distance to multiple centers, as I mentioned before. And the important thing here is that depends heavily on the selection of the correct k-value. Okay, so if you don't choose the right k-value, the interpretation is going to be very different. When your data is not good in ways that is not homogeneous, and you will see that it performs poorly. You have density-based algorithms, and I'm not going to go into much. Basically use density to connect groups. And the good part of this is that it can capture more complex structures, like low-dimensional data, but doesn't tend to find clustering uniform in large data. So density-based algorithms are being used now, for example, to do the clustering for single cells. Single cells need to use clustering. So yeah, density algorithms is one of these common algorithms that are being used now. And then you have a statistical method like cell organizing maps or Gaussian mixture models. And also, well, as I said, you have some statistical method that you apply, and there can be very different between them. Basically, say that they have limited success and non-normally distributed data. So even if you have some opportunity to use those for your RNASeq data, they are not common, okay? Because, as I said, if you have non-normally distributed data, may not be the best. I like this graph that I found also in the paper, because from one hand, it defines how these algorithms, different algorithms will behave in different types of data. No? So you apply k-means, WAR, you remember this is the hierarchical, density or mixed models. If you have non-structural data, free clear cluster, 2 wide clusters, 2 half-moon clusters, or 2 nested, okay? So this is how will these algorithms will classify those. No? So for example, by k-means, you have free cluster. Well, you know that you have free cluster, you put free cluster, and it's perfect. Meanwhile, with WAR, hierarchical clustering, probably they are going to put these two in the same, and then you will have this one. And if you scan, will check for will scan for density. So the points in the center that are very dense will have a different cluster than the points outside. When you start in the density, will start to change. A mixture model, as I said, it will depend. So this is a good example how these things behave from one place to another. Again, we use more or less these two for RNASeq data. Of course, once you have the clusters, when you do clustering, you can use different methodologies to evaluate how good are the clusters. So you can check for biological relevance. That is the most important one, of course, always. Then stability. So when you change case, for example, how this clustering behaves. Cluster fitness, that can be a combination of different parameters. Okay, so you also, as I say here, no, it's the accounting for noise and uncertainty is important in order to interpret these clustering results. You have different ways to test clustering. Again, this could be a course. I saw, for example, silhouette wild is one of them, gap statistics is another one, F statistics. So you have all the different measures. So for example, to measure compactness, you use the RNASeq. So you have different ways to evaluate clustering. And I'm not going to go into that because that will be too much, okay? But just keep in mind that also you can evaluate clustering. How important is this? Let me tell you, it's very simple. Right now, when you are doing a really simple RNASeq analysis and when you are comparing conditions, you may run just one of these clusters, and it more or less gives you the results that you wanted. It's fine. You don't need to move forward. But especially because clustering is essential for single cell, some of these evaluation methods is something that you need to apply after that you are doing clustering with single cell in order to really verify that you are able to assign cluster to the cell types, because that is one of the main topics, the main reasons of single cell. So these evaluation are very important there. Yeah, sorry, this is a mistake. So tools for performing RNASeq analysis. So most of the tools to perform this are in R packages. You want to have a really good summary of the different clustering methods also with R, not applying to RNASeq specifically, but just for R. That is a really good page. So we'll talk about partitioning methods like k-means, hierarchical clustering, fuzzy clustering, density, and model-based. So that is a good part. And in terms for us, we can play just with these tools that I have in this table that are the most common ones used for RNA-seq analysis. So you can, usually you use distance from stats to calculate the distances. We already did that. And then hclust to do hierarchical clustering, or k-means to perform k-mean clustering. Then another important tool is this FPS package, because you have methods to compare clusters and optimize clustering. PV clustering is a different package and this is interesting because also can perform different hierarchical clustering and assign different p-values. And then if you want to run model-based clustering, you can use MBClusterSeq for RNA-seq. Or if you want to do this model, well, another model clustering is self-organizing map, and you can use the sum. I would say that this is not, I never saw this using, so the most common one for model-based clustering will be this MB cluster. So the other last thing that I want to tell you is about the hierarchical clustering. As I mentioned before, it gives you the hierarchical relationships as you can see here. But one thing that you can do with hierarchical clustering is you can, once you do the hierarchical clustering, you can define where you want to cut. Okay, so you can say, okay, I want to have 5 groups, and then you have a function that it will scan the tree until you find 5 groups. No? So for example, in this case, 1, 2, 3, 4, and 5. So you will have 5 groups here. So it will cut the tree here and will give you all the different groups. So this will be one group. This will be another one, another one, another one, and another one. Okay, so that is an important concept. And of course, you also can visualize the cluster, and you will do in the exercise using ggplot. No? So that is a very useful part too. Okay, questions that you may have about clustering. No questions? Are you still there?
Yep.
Okay, perfect. Okay. Of course, I'm going to propose you to do an exercise because I think that is interesting, and of course, is the funny part of this course. So before I stop it, so let me stop it the recording now and start a new one for the next part. I need to, don't forget, stop the recording and